Hello, everyone. Our lesson for today is about microbial respiration. Microorganisms they display vast evolutionary divergence and exhibit exceptional metabolic characteristics. This mechanism allows the conversion of sugar into energy and regulates its storage and release for different cellular processes. So in this lesson, we will learn the basic concepts and some of the various metabolic processes in microorganisms. And we will be highlighting microbial respiration. The end of this lesson, you will be able to explain the Krebs cycle and its importance, compare anaerobic and aerobic respiration, and discuss the process and concepts of fermentation. The motivation question for this lesson is, why do you think glucose is important in a biological system? Okay, I hope you'll be able to answer that one at the end of our lesson. So the general concept no, in energy generation no, is illustrated in this diagram. So we know that light in terrestrial environment or in aquatic environment at the photic zone where light can still penetrate in the aquatic environment. Now, light is the ultimate source of energy, and this enables primary producers like the photosynthetic organisms to undergo photosynthesis. In this process, ATP is generated now, and sugars no, and or other polysaccharides are generated as well. Now, so during the light reaction of photosynthesis, ATP is produced, and this is the one that is being used for the dark reactions. We will be uh, discussing more about photosynthesis when we go to or for our next lesson. Now, these polysaccharides that are formed via photosynthesis could be used for the generation of ATP. So it could be directly phosphorylate. This could be directly used to generate ATP through substrate level phosphorylation, and it could be consumed by heterotrophic organism as a source of their energy. So this light energy will be stored in the form of ATP. And in order to generate this ATP, there is a high diversity of energy generation in microbes. For us humans and even in plants, we only have one way to generate ATP. But for microorganisms, now they have a diversity. They have different types of energy generation. Now, energy generation or respiration is it's an enzymatic reaction. So these are catalyzed by enzymes. We know that enzymes are proteins that serve as catalysts. So when we say catalyst, we're talking about those substances that speed up the rate of a particular reaction. They are not consumed in the reaction. Okay. So they serve as catalysts for several reactions to convert molecules that we call the substrate into products that can be used by the cell. So if you have here an enzyme, this is the substrate to where they act upon. There is an active site where the enzymatic catalytic activity occurs, and in the process, after the catalytic reaction, then the products are produced. So this time, this is a decomposition reaction. One substrate is turned into two different products. No? A catalytic reaction could be reversed as well. No, it could be two products as to be synthesized into one, or two substrate to be synthesized into one product. There are also what we call cofactors. These are non-protein molecules that are required to make the enzyme catalyzed reactions work. So they are either inorganic or organic. So they could be called coenzymes as well. Inorganic cofactors are those metal ions that increases the rate of catalysis between the enzyme and the substrate. So these are either attached or loosely bound into the enzymes. And coenzymes are organic cofactors that aid in enzyme catalysis by binding loosely in the enzyme active site, thus enhancing substrate binding. So examples of these coenzymes include FAD or flavin adenine dinucleotide, nicotinamide adenine dinucleotide, and nicotinamide adenine dinucleotide phosphate. So all living cells need energy to move and grow. The energy to build cells comes from chemical reactions. So the sum of all the reaction that occurs in living organism is what we call metabolism. There are two types of metabolism. It could be catabolism or anabolism. So when you say catabolism, you're talking about the breakdown of complex molecules into smaller ones. So breakdown, catabolism. But if it is building up, no, so when you build something from several substrate, then that is what we call anabolism. So these are reactions that build the cells. So in catabolism, there is provision of energy for anabolism. However, some of the energy is released as heat. That's why energy storage is as we go along the various process now of the reaction, the energy, some of this will be transformed now as heat, although they are never lost. Okay, so there are different types of energy carrying molecules, and the most known and the readily available molecule is the ATP. So adenosine triphosphate. Okay, so it is composed of adenosine diphosphate plus an inorganic phosphate. So it is composed of three. It is basically a nucleoside or a nucleotide with two phosphate groups. However, the last phosphate group is actually an inorganic phosphate. Okay, so this is like the energy currency of the cell. This could be and readily be used, to fuel some of the reactions. So this is the major carrier of chemical energy, and they can transfer energy to cell processes in three different ways. So since there are three phosphate group attached, to this nucleoside, the yeah. So first way that you could harness the energy is through hydrolysis that release inorganic phosphate, or you could hydrolyze, or break down the ATP that would release 2 phosphate. We call it the pyrophosphate. And you could phosphorylate an organic molecule. So besides ATP, other nucleotides carry energy as well. For example, guanosine triphosphate that provides energy for protein synthesis. Now, another form of energy-carrying molecules are the NADH. Now, although unlike ATP, NADH is cannot be readily be used. They carry a lot of energy, but they cannot be used directly. They need to be transformed into an ATP first. Okay, so nicotinamide adenine dinucleotide carries 2 or 3 times as much energy as ATP. It also donates and accepts electrons. NADH is the reduced form, and NAD plus is the oxidized form. You already know when it is reduced, and you already know when it is oxidized. So reduced when it has, or when it has accepted, an electron, oxidized when it has lost, an electron. The overall reduction of NAD plus consumes 2 hydrogen atoms to make NADH. So NAD plus 2 protons plus 2 electrons equals 2 NADH plus NADH plus protons, and the Gibbs free energy is 62 kilojoules per mole. So this reaction requires energy input from food molecules. So if we can produce, if we can utilize energy in 3 different ways, there are also 3 ways in order to generate, how to produce ATP in the bacterial and archaeal cell. No? And even in eukaryotic system, Now, these are also possible. So, first is through substrate level phosphorylation. So, phosphorylation is the production of ATP. So, it could be through substrate level. So, there is transfer of high energy phosphate from phosphorylated organic compound. Or it could be respiration linked phosphorylation, so production of ATP by oxidation of a reduced organic or inorganic compound. So this compound is the electron donor and coupled with the reduction of an electron acceptor. So there will be generation of a memory-associated proton motive force, which we will discuss in detail later. And the third type is through photophosphorylation or photon-linked phosphorylation. This occurs during the light reactions of photosynthesis. So light energy is used not to give electrons and generate the proton motive force that is necessary for ATP production. So as we've said, ATP is a universal medium of biochemical energy exchange, and living organisms can establish a charge separation across the membrane. That's why chemosynthesis is or the production of ATP via the charge separation through the ion gradient is important to establish an electrochemical potential. This is because protons are extruded through the membrane. The electrochemical potential can be converted to ATP. Okay, ATP can also generate the electrochemical potential. So the electrochemical potential can be used to transport nutrients. NAD plus or NADP plus are sources of protons and electrons, or the reducing power. Okay, so this is what we call the proton motive force for ATP generation. So what is happening here is, as an electron is donated into the system, okay, these are the electron acceptors embedded in the membrane of respiring microorganisms, or if in eukaryotic system, these are found in the inner membrane of the mitochondria. So for respiring microorganisms, these are found at the membrane, respiring microorganisms. So they will accept electron from electron donor. So it could be NADH or NADPH or FADH2. Now, as electrons are transferred in these different electron acceptors, protons are extruded outside. Okay. Now, as more electrons are donated, more and more protons are extruded, no, into the periplasm, and this creates high concentration of protons outside. Okay, so there will be charge separation. So what happens is that because of the high concentration of protons here, more of the protons will tend to go back inside. How do they do it? They pass via an ATP synthase. Now, as the protons are accepted here, that the protons serve as fuel, in order for the ATP synthase to convert this electrochemical potential into an energy that will be stored in an ATP. So an adenosine diphosphate plus inorganic phosphate will be fused or will be synthesized into an ATP via the protons that are passing through the ATP synthase that drive this synthesis. So this is what we actually known as the proton motive force. This is the respiration part. Why is it called respiration? Because as electrons are transported here, in order for more electrons to be accepted by the electron acceptors, the electrons that lost its energy must be accepted by an electron acceptor here. So in aerobic respiration, the oxygen serves as the final electron acceptor to remove the electron that has already lost its energy. So you will have, as these protons also enter, so you have 1/2 oxygen plus 2 protons plus 2 electrons will give you water. Okay, so this is another representation of the charge separation that is created an electrochemical gradient in the proton motif force. Okay, so now let us specifically talk about carbohydrate catabolism, or the sugar catabolism, or the breakdown of sugar to produce energy. So there is transfer of phosphate from a phosphorylated carbon substrate to adenosine diphosphate, and this occurs during breakdown of sugars. We call it glycolysis. While if and under anaerobic conditions, the breakdown of molecules could be through fermentation. So chemotrophic microorganisms, the electrons are extracted from inorganic molecules like H2, to produce two protons plus two electrons. For heterotrophic organisms, energy is stored in carbohydrates, and electrons are extracted from organic molecules like lactate to produce pyruvate, two protons and two electrons. So glycolysis is actually the breakdown of uh, glucose. There is substrate level phosphorylation, meaning the production of ATP at the substrate level. So what happens is that glucose is first activated, and undergoes molecular rearrangement via the phosphorylation of 2 ATP. It becomes fructose 1,6 bisphosphate, which is highly unstable molecule, and so it becomes 2 glyceraldehyde 3 phosphate. So glyceraldehyde 3 phosphate, meaning it is a 3 carbon sugar which phosphate attached to it's the third carbon. So 2 glyceraldehyde 3 phosphate is acted upon by 2 NADH, and NAD plus will be reduced into 2 NADH, as it takes in the uh, electrons, the protons. Okay, 2 inorganic phosphate is also attached into the glyceraldehyde 3 phosphate, making it 1,3 bisphosphoglycerate. So 2 of this, because there are 2 already, and for each one it has 2 phosphate attached, then an ADP, adenosine diphosphate, 4 adenosine diphosphate will take out those 4 2 from each of the bisphosphoglycerate to produce 2 ATPs for each. So that makes 4 ATPs all in all. And this will be oxidized to form pyruvate. Okay? So one glucose could later on produce 2 NADH, 4 ATPs, and 2 pyruvate or pyruvic acid. However, there is an initial expenditure of 2 ATP at the start during glucose activation, and so there is only a net gain of 2 ATP per glucose molecule. So we are talking here of one glucose molecule that has undergone glycolysis. So what happens next? Now, in microorganisms, for higher eukaryotic organisms, this is always true. Glycolysis always occur. But there are also in microorganisms, the glycolysis could also be called Embden-Meyerhof pathway. Although there is a particular portion here when the Embden-Meyerhof pathway, or the EMP, is slightly different from glycolysis. So this is the major route of glucose catabolism in many aerobic microorganisms. In this process, phosphate is transferred from an energy-rich molecule to ADP. And in some anaerobic microorganisms, this is the sole pathway for generating ATP via fermentation. So just like in the glycolysis, so bisphosphoglycerate now becomes phosphoglycerate, and through these enzymes, it will produce pyruvate, and this pyruvate will now enter the tricarboxylic acid cycle. So in the EMP pathway, a glucose molecule undergoes a stepwise breakdown to produce 2 pyruvate molecule. Now, each pyruvate is a 3 carbon sugar. Now, starting with a 6 carbon sugar na glucose. So this is the detailed reaction. So just like glycolysis, glucose is first activated to produce glucose 6 phosphate, fructose 6 phosphate, and phosphorylation will become fructose 6-bisphosphate. Now, in glycolysis, the fructose 1,6-bisphosphate will be broken down into two molecules of glyceraldehyde 3-phosphate. However, in EMP, one of the three carbon sugar becomes dihydroxyacetone phosphate first. So one becomes like glyceraldehyde 3-phosphate, while the other becomes dihydroxyacetone phosphate, and that makes it different from the original glycolysis that occurs in other organisms. However, dihydroxyacetone phosphate could be converted into glyceraldehyde 3-phosphate via the triphosphate isomerase enzyme. And so the reaction will occur. There's production of NADP, NADH, and ATP in the process, producing also pyruvate. Now, there are also alternative glycolytic pathways that is believed to have evolved earlier than the EMP. So glucose in Itner-Dudorov pathway, or the EGP, glucose is activated by one phosphorylation reaction and then dehydrogenated to 6-phosphogluconate. So it is then dehydrated and cleaved to pyruvate and glyceraldehyde phosphate that enters the EMP to form pyruvate. So the ED pathway produces one ADP, one NADH, and one NADPH. If you compare, aside from the pyruvate that is also produced. So if you compare that to the EMP produces a net yield of 2 ATP and 2 NADH, but it does not produce NADPH. Okay, so this is the Entner-Dudorov pathway. So the starting molecule is still a 6-carbon glucose. Now at the end, it will produce this oxodeoxygluconate, which will become glyceraldehyde 3-phosphate and pyruvate. So this glyceraldehyde phosphate will undergo a glycolysis also to become pyruvate. Okay. So another alternative glycolytic pathways is the pentose phosphate shunt pathway. So the PPS is like the ED pathway. So the pentose phosphate shunt forms 6-phosphogluconate. So this is the 6-phosphogluconate now in 6-phosphogluconate in the ED pathway. And here, pentose phosphate shunt also produces that, which is then converted to the key intermediate product, which is the ribulose 5-phosphate, which in turn produces a series of sugars, each containing 3 to 7 carbons. Now, this pathway produces for the one glucose molecule that undergoes pentose phosphate shunt pathway, it will produce 1 ATP and 2 NADPH, no NADH. If you compare that to EDP will produce 1 ATP, 1 NADH, and 1 NADPH. In here, there is no NADH, but there are 2 NADPH. Okay, so this is the detail of the reaction. Now, so again, it also produces 6-phosphogluconate, but then this is converted into the key intermediate product, which is the ribulose 5-phosphate, which then becomes the which produces the pyruvate in the process. So the production of ATP via the glycolysis and other glycolytic pathway is what we call the substrate level phosphorylation, because upon the breakdown of the substrate itself, ATPs are produced. But the second type of ATP generation is through respiration-linked phosphorylation. So how do ATP, how do microorganisms produce ATP via respiration? So the first type of respiration is what we call aerobic respiration. So this is a process done by organisms that utilize oxygen for energy production. So they are the aerobes, no? Because aerobic, so meaning there is oxygen involved. So after glycolysis, we know that pyruvic acid or the pyruvate are produced. So pyruvate is utilized by aerobic organisms to produce energy through the citric acid cycle, the Krebs cycle, or the TCA, tricarboxylic acid cycle. So the TCA cycle is also known as the Krebs of the citric acid cycle. In prokaryotes, it occurs in the cytoplasm. However, in eukaryotes, it occurs in the mitochondria, and we know that mitochondria has evolved from a proteobacterium. Glucose catabolism connects with the TCA cycle through pyruvate breakdown to acetyl-CoA and carbon dioxide. So acetyl-CoA then enters the TCA cycle by condensing with the 4-carbon oxaloacetate to form citrate. So this is the structure of an oxaloacetate. So before after the formation of pyruvate, after glycolysis, the pyruvate is first converted into an acetyl CoA. In the process, NADH are also formed. Then the acetyl CoA, so you have here the pyruvate, which is a 3-carbon sugar. Now it will lose a carbon, so carbon dioxide through a carbon dioxide, and it will be oxidized by NAD plus. NAD plus will be reduced into NADH. So for every molecule of pyruvate, 1 carbon dioxide and 1 NADH is formed. So a 3-carbon pyruvate will now become an acetyl CoA, which is 2 carbon because 1 carbon dioxide, 1 carbon has already been lost. Okay. So the acetyl CoA is then accepted by a 4-carbon oxaloacetate to form a 6-carbon citrate or citric acid. That's why it's called citric acid cycle, because the first intermediate product is the citric acid. So it will then be oxidized by NAD plus to produce 2-oxoglutarate. In the process, carbon dioxide is lost, and so it will become a 5-carbon sugar. Again, it will be oxidized, producing NADH in the process, and also losing carbon through its carbon dioxide, becomes succinyl-CoA 4-carbon sugar. Okay, succinyl-CoA will be dephosphorylated. It will be further oxidized to form oxaloacetate. In the process, ATP is formed, NADH and FADH2. FADH2 or flavin adenine dinucleotide is another form of an energy carrier molecule like the NADH. So for 1 pyruvic acid, 3 carbon dioxide are formed. Okay. You have 1, 2, 3, 4 NADH are formed, and 1 FADH2 are formed, and 1 ATP. Since there are 2 pyruvic acid that's been produced by glycolysis from 1 glucose molecule, so you can just multiply by 2. Okay? So you have here, during the TCA cycle, there are 3 NADH formed, 1 FADH2 and 1 ATP, or it could be GTP. But since 2 pyruvate are formed, then you can multiply this by 2. So for 1 glucose molecule that enters TCA cycle, 6 NADH are formed, 1 FADH2 and 1 ATP. Okay? So that reaction producing ATP via the tricarboxylic acid cycle is also a form of substrate level phosphorylation. Okay? So now what happens? So the energy that has been stored in NADH and FADH2 will now be harnessed via the electron transport system. So in the electron transport system, the NADH that has been formed will be donating the electrons into the ETS here. So during the glycolysis, the 2 pyruvate molecules and 2 NADH and 4 electron molecules, 2 NADH are formed. So 2 NADH will donate 4 electrons to the ATS. During the formation of the pyruvate, 2 NADH are also formed. So it could also donate 4 electrons here to the ATS. And during the TCA cycle, there were 6 NADH formed. So it could donate 16 electrons. There are also 2 FADH2 formed. Okay? So for each NADH, no, and in here in the ATS, just like we discussed earlier, this will, as the electrons are donated here, protons are also extruded out, and so the proton motive force will be activated, producing ATP in the process. Okay. So for each NADH, there will be 3 ATPs formed, and for each FADH2, there will be 2 ATPs formed. So if there are 6 NADH, 2 NADH, and 2 NADH, that means 10 NADH, no, for one glucose molecules. Then you already have 30 ATPs formed from NADH alone. For each FADH2, there are 2 ATPs, so 2 times 2 will give you 4 ATPs. So for one glucose molecule, in the ATS, there will be 34 ATPs produced. But if you add the 2 ATPs formed substrate level phosphorylation during the TCA cycle and during the glycolysis and net yield of 2 ATPs, then that makes 38 ATPs now produced for one glucose molecule. So this here, production of ATP via the proton motive force in the ATS is what we call respiration-linked phosphorylation. Okay? So these are substrate level phosphorylation production of ATP, and this is respiration-linked phosphorylation. In the next lesson, we will be talking about photon-linked phosphorylation, or during the photophosphorylation during the photosynthesis process. Okay, so it is just been talking about electron transport chain. So there is movement of the electrons via the ATS. So there are a series of electron donor and electron acceptor. So this is still the same, just like what we talked earlier. So in order for more electrons to be donated here, a final electron acceptor must accept this electron para mawalan yung electron here. So 2 free hydrogen ions plus 2 electrons exiting ETC plus 1 of an oxygen molecule as the final electron acceptor will produce water. Okay? Now, this is the summary of the reactions for glucose metabolism or glucose catabolism. Okay? So, katong giingon na ako earlier. Now, in other organisms, just like anaerobic microorganisms, since they do not consume oxygen because oxygen is toxic to them, how do they they perform the respiration link phosphorylation, since dili man sila kon oxygen, and oxygen is very vital in the respiration linked phosphorylation. Without oxygen, all of these cannot proceed. So for microorganism, and this is only unique to prokaryotes, they usually possess alternative electron donors and electron acceptors. Okay, so electron donors could be formate, hydrogen, NADH, lactate, or succinate, and these could be alternate substrate oxidoreductases. Okay, so terminal electron acceptors in replacement for oxygen in anaerobic respiration could be nitrate, nitrite, thiosulfate, and fumarate. Okay, so these are the different forms of oxidized nitrogen. So nitrate is successively reduced as follows, and sulfur as well. Okay, Now, so that was true for anaerobic respiration. Now, so it is just a respiration without the use of oxygen and using or utilizing alternative electron donors and alternative final or terminal electron acceptors. Now, there are also those microorganisms that do not really undergo tricarboxylic acid cycle and electron transport chain. Okay, so the only source of their energy is through glycolysis alone. Okay, so we call this fermentation. So this is a sum of anaerobic catabolic reaction. ATP is generated only by substrate level phosphorylation. No, so wala sila electron transport chain or respiration link phosphorylation. No electron acceptor is needed. No, so just which is very different from aerobic and anaerobic respiration. And there is an incomplete oxidation of a substrate to alcohols or acids. Fermentation is a balanced reaction. No, ang pinaka-importante lang is to regenerate na NAD. Fermentation could be done on sugars or amino acid. So the very common examples are the alcoholic fermentations performed by yeast and lactic acid fermentations performed by lactic acid bacteria. Now, there are different types of fermentation. It could be alcoholic, homolactic, heterolactic, butyric acid, butyrial, and mixed acid. And these are the different products. So say for example, for alcoholic fermentation, they produce ethanol plus carbon dioxide. For homolactic, lactate, heterolactic, lactate plus ethanol plus carbon dioxide, and so on and so forth. And these are the examples of microorganisms that undergoes this different type of fermentation. So say for example, Zymomonas mobilis and Saccharomyces cerevisiae will produce ethanol plus carbon dioxide through alcoholic fermentation, and so on and so forth. So the very common type of fermentation is the homolactic and alcoholic fermentation. So in this process, glycolysis still occurs. The glucose is broken down to 2 pyruvic acid, producing a net yield of 2 ATP and 2 NADH. Now we know that in aerobic respiration, pyruvic acid must be converted into acetyl-CoA, then tricarboxylic acid and ETS. But since there is no oxygen, so the microorganism does not do that anymore. Now the important thing here, in order for a glucose to be converted into pyruvic acid, is that there is a reduction of NAD plus to NADH. Okay? So the important thing here is that NADH must be converted back to NAD+. So it must be regenerated so that glycolysis could still occur and energy could still be produced. Okay? So how does the microorganism do that? Since they do not do the ATS, in aerobic microorganisms or those microorganisms that perform respiration, NADH could only be regenerated to NAD+ via the ATS because they will donate their electrons there. Since for fermentative microorganisms they do not do that, someone must accept the electrons from NADH in order for NAD+ to be regenerated, so for the glycolysis can still occur. So how does it do that? Okay. So in lactic acid fermentation, the pyruvic acid will act as the electron acceptor for the regeneration of NAD+. So pyruvic acid plus the electrons from NADH losing 2 protons in the process will give you 2 NAD+, that will be converted to lactic acid. Or for alcoholic fermentation, 2 pyruvic acid will lose 2 carbon dioxide to become 2 acetaldehyde. The acetaldehyde will become the electron acceptor of the NADH, losing 2 protons and becoming 2 NAD+ in the process and converting acetaldehyde to ethanol. So this is lactic acid and this is alcoholic fermentation. Okay, so again, these are the different types of glucose fermentation. So using glucose known as the substrate for the fermentation. But there are also fermentation that uses different types of substrate. No substrates other than sugar. So say for example, glycine plus alanine. So these are amino acids. So these are what we call this is what we call stickland fermentation. In the process, acetate is produced. Now the organism uh, that would perform this is the Clostridium sporogenes. Another type is propionic acid fermentation. So using lactate, it could produce propionate plus acetate. Acetylene fermentation, oxalate fermentation, malonate, and homoacidic acid fermentation. These are the overall reaction and the type of organisms or the example of organisms that perform such type of fermentation. Okay, so in order for you to review all the things that we have discussed, there are quite a lot. So I am providing you these summary questions. So what is Krebs cycle and why is it important? How does aerobic respiration different from anaerobic respiration? What is fermentation? What are different types of fermentation? What are the different types of glycolysis and some modifications to alternative glycolytic pathways? So these are the things that you should remember in this lesson. So I would like to acknowledge Miss Maria Sherlita S. Rosal for the help in making the slides that I am using now for the presentation. If you have questions regarding the lesson, you can contact me through my email or your respective lecturer for their provided email addresses, or you can directly key in your questions via the Facebook Messenger group chat. So thank you very much and see you again in our next lesson.